Hey man, how's it going? Hey, can you hear me?
Yeah, now I can. Hey,
Yo,
alright, so I sent you the chapter 9.
okay. 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 So why don't you open up the exam, the one that doesn't have the answers,
Okay,
and yeah, we can go through these.
alright, 1 second.
Oh, I got the chapter 9 thing. Is this on your test? Chapter 9?
Yes.
Okay. All right, I guess we can go over chapter 9, the slides first.
Okay. 1 second.
In situ.
Okay. Alright, so we'll go over this.
Mm-hmm. Yep. So it starts off with the genome, which you pretty much know what that is. And how to hybridize. So yeah, in the slides that talk about heating the DNA double helices, causing denaturation, which breaks it into the single strands. And then when you slowly cool it, double helices reform. And that's just from the regular hydrogen bonds. And then where it says DNA can undergo reversible strand separation, that's pretty much another example of it, but instead of just heat, you can also use a strong base. That's what the OH minus is, the hydroxide. That's what that is. So, you have native state and single-stranded denatured state. And then you have essentially, it essentially forming those bonds again in the renatured state. And then fish. So, just from looking at the practice test real quick, it seems like they asked some questions about it, so this is important to know. It means fluorescence in situ. I think that's how you pronounce it, right? Let's see.
In situ.
Yeah, fluorescence in situ hybridization. That's what FISH stands for.
Yeah.
So essentially you have chromosomes, which, you know, are only chromosomes like during mitosis. And, you know, they're on a glass slide. You treat the slide with a hot salt solution to denature the DNA. So then you, like, you know, from the like zoomed out view, it looks the same, but what you really are doing is you're taking the double-stranded DNA and you're making it single-stranded. And then you incubate it with biotinylated DNA, biotinylated DNA probes. And essentially what that is you take biotin and you attach it, you covalently attach it to the nucleic acid, essentially.
Okay.
And what that does is it acts as a label.
I see.
Okay, so that acts as a label, and then you wash it to remove the unhybridized DNA. So in this example, satellite DNA is employed. Let's see what that is. Satellite DNA. And satellite DNA is basically very large, consists of very large arrays of tandemly repeating non-coding DNA.
Okay.
Okay, and then you incubate it with fluorescently labeled Avidin. And Avidin is a glycoprotein that combines specifically with biotin.
That specifically binds to biotin?
Yeah.
And biotinylated again, just to go over meant what again?
It's just it's like a vitamin that you attach onto the DNA. Here, I'm gonna open up Microsoft Whiteboard and share my screen. So I'm gonna draw it out for you. Okay, you can see my screen.
Mm-hmm.
Okay, so basically what's happening is Well, okay, so let's see. Let's see if I can do this. Okay, so biotinylated basically means that we're adding a biotin group. Basically, like, let's see if I can find a better picture. Well, okay, so basically what you're doing is for the single-stranded DNA, you add biotin to it. So yeah, this is just basically adding the biotin to it, and then you wash away the. So okay, then you wash, and then hold on, and then, well, these are like the strands that are not biotinylated, and you wash to remove the non-biotinylated strands. So you know, you're basically just left with the biotinylated ones.
Mm.
And then you incubate it with this thing called Avidin, which fluoresces, so it basically, you know, gives off, like, you know, light, like a color, and that binds to the biotin.
Mm.
So looks like that right there. So basically, you know, you add biotin to the single-stranded DNA, you wash it to remove the non-biotinylated DNA to just get the biotinylated DNA over here, and then you add Avidin, which is a fluorescent probe, and that binds to the biotin on the biotinylated DNA.
Yeah, and then the counter stain.
Let's see, counterstain chromosomes, so they appear red. Okay, localization of satellite DNA in the centromeres. Okay.
Yeah, so you can tell where the chromosomes are, right?
Yeah, I believe so. Let's see. Satellite DNA. It consists of very large arrays of tandemly repeating non-coding DNA. It's the main component of functional centromeres and form the main structural constituent of heterochromatin. So this image over here kind of shows you that like the centromere contains the satellite DNA, so do the telomeres, because they're essentially, you know, they don't code for anything.
Yeah.
Yeah. let's say satellite DNA, non-coding, located in telomeres and centromeres. Okay. So here we have fish of satellite DNA. So you essentially have the fluorescing yellow parts in what appears to be like the centromeres of these chromosomes.
Yeah.
Yeah. Now, let's see. Chromosomal localization of a non-repeated DNA sequence. So let's see. For a gene that encodes the nuclear lamin protein, it's present on 2 of the homologs of chromosome 10. And then they're asking, why are there 4 dots? 4 dots. I guess they mean this, 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 and this. Well, I guess because they're homologous. Wait, 4 dots. Let's see. 1, 2, 3, 4. I mean, if they're homologous chromosomes, it should be on. I mean, they should have. It should be double the amount, right?
Yeah.
Okay. DNA sequence variation. Any 2 humans have about 99.9% similar nucleotide sequence. Only around 1 out of every 1,000 nucleotides is different. These differences are mainly single nucleotide changes, for example, an A for a G. Due to a mutation, those are single nucleotide polymorphisms, SNPs. Of the total 3 million SNPs, only 60,000 are found in protein coding regions or sequences that lead to different amino acids. These amino acid differences may change the function of the protein, thus we all have different phenotypes, aka look different. So, you see people looking different over there. Genes and genomes can be altered by several mechanisms. So we have original genome here, and then altered genome here. So this, can be altered by a mutation within a gene. So, the gene is the orange part, then that red part, the mutations inside of it. It can be a mutation in regulatory DNA, so some part that is not part of the gene, but it's part of like a regulatory region, like promoter or enhancer or something like that. And then exon shuffling, aka alternative splicing and stuff. So they could be different order of the exons. And then there's transposition. So I saw some questions on the test about that. So they'll probably talk about it in these slides. So basically, it's part of the genome that is mobile, like it can move, it can be transposed. So we have this purple part, which is the mobile genetic element, and it gets inserted into this regulatory DNA region over here. And there's also horizontal transfer, which is from one organism to another. So it seems like this red gene over here gets transferred to organism B. So organism B has a new gene from organism A. You see that in bacteria with something called transformation.
Yeah, we talked about that.
Okay, now it seems like they're going to go more in depth with each of these things. So for the mutation within a gene, it may help or hurt the organism, but most of the time it's neutral. So, within an intron, or it'll produce the same amino acid. If it produces the same amino acid, it's called a missense mutation. And there's synonymous mutation, aka known as a silent mutation. So it's a mutation that essentially will lead to the same polypeptide. And, that's very, likely because, remember, like, the wobble thing? Like, so many different codons can code for the same amino acid. So that's a silent mutation, also known as a synonymous mutation. And then there's a non-synonymous mutation, which is when a different polypeptide is produced. Does that make sense?
Yeah.
All right. Now, mutations in germline and somatic cells are different. So, essentially what we have here is, so I guess we have two germ cells, aka, like sex cells. That's why they're drawn as like a semicircle. And they combine to make the zygote, which, makes the organism that makes the somatic cells, the, non-sex cells. But it also makes, the germline cells, which move on to the offspring. So essentially that's what they're kind of showing here. Now, now they're saying A and B are mutations. A is a mutation in the germ cell line, and B is a mutation in the somatic cell line. So, if the mutation's in the somatic cells, it's only going to affect that one organism. But if the mutation is in the germ line, it's going to affect the offspring. So it basically carries over.
Okay.
Yeah.
So repeat that again, if a mutation is in the...
Yeah, so it's like if you have a mutation in any cell in your body that isn't a sex cell, it's only going to affect you. But if it's in your, like, sperm, then it's going to affect your offspring. It's going to affect your kids. Because it's going to be passed on to them.
Okay, so if the mutation is within the somatic cells,
Mm-hmm.
If a mutation occurs in the somatic cells,
Mm-hmm.
that means what?
That means it'll just affect that that individual, because it's in the body. You know, it's in a cell that's responsible for things in the body. But if it's in a sex cell, it's going to affect the offspring.
If it's within the germline?
Yeah, it will affect the offspring.
So if it happens in the somatic cells, that means it'll just occur in the individual because it is in the cell responsible for the body?
Yeah, just because it's in a cell that is not responsible for, offspring, for kids, basically. Like a germ cell is just a sex cell. It's like an egg cell or a sperm cell. So like if that has a mutation, then that's going to affect the offspring, right? The children.
Yeah.
Yeah. But if it's just in a non-sex cell, aka a somatic cell, then it's just going to affect that particular individual, but it's not going to be passed down.
Got it. Okay.
All right. Okay, now mutation rates can be measured in the laboratory.
Okay.
So let's say we have a mutant E. coli cell that requires histidine, which is an amino acid, to proliferate. It needs that amino acid in order for it to make more of itself.
Mm-hmm.
And the particular mutation is in, so it's in this gene over here. So we have an inactive, histidine gene, which is this, and it produces this mRNA, and which does a premature stop codon. And that's also called the nonsense mutation. And what that does is it eliminates an enzyme that's required to make histidine. So this E. coli cell basically cannot make histidine itself. And then what they do is they put it into, they make a culture, and they put it into a medium that has a lot of histidine in it. So it allows all of the bacteria to multiply, to make more of itself, because it has the histidine that it's not capable of producing itself.
Mm-hmm.
And then as the cells divide, random mutations arise spontaneously. So, zooming in on this, you see bacteria in which different mutations have occurred, like this blue one and this red one over here. And then you can take a sample of the cells that are spread on the petri dish. and you can put it into a medium which lacks histidine. So basically, that means that since we're starting off with bacteria that cannot make histidine, and we let it proliferate in a medium that has histidine, and make mutations, then when you put it back into a medium that does not have histidine, you'll have one of those mutations from this step that occurred that allows it to proliferate in the absence of histidine. So there basically is a new mutation that formed which restores the production of the enzyme required to make histidine. And this is a way for them to measure the rate of mutation. And they essentially found that there's going to be 3 mistakes per 10 to the 10th power, which is 10, billion, I believe. Yeah. Or rather, 100 billion. So there's basically 3 mistakes per 100 billion nucleotides that are copied. The mutation rate is based on the size of the genome and the number of bacteria in overnight growth. Statistically, every single non-lethal base pair substitution is present in the culture. For humans, there's only going to be 1 mistake per 100 billion nucleotides. That makes sense?
Mm-hmm.
Alright, now for humans, fertilized egg to female gamete requires, basically undergoes, in that process for it to go from a fertilized egg to another, unfertilized egg, right? It takes 20 to 30 genome replications. So basically it undergoes, whatever, cell division, that many times. Fertilized egg to sperm requires around 200 genome replications. So, 10 times as much. And there's about 0.1 to 1 mutations per replication, which is also going to be 10 to 100 mutations between the individual gametes that they make. So essentially, 2 germ cells make the zygote, which is the fertilized egg. And it takes 20 to 200, depending on if it's going to make an egg or a sperm, to go from zygote to making more germline cells, to making more sex cells. You good? Okay. And now they'll talk about mutation in regulatory DNA. So mutations can change the regulation of a gene. Oh, this is interesting. Percentage of the population that is lactose tolerant, meaning that they can digest milk, the sugar in milk.
Mm-hmm.
And you see it's pretty high in these kind of regions. These kind of northern latitudes. But there's a lot of no data, but in this kind of middle like latitude over here, it goes down to like 50%. In China, it's like 0 to 9%. In India, it's higher than in China, which is interesting. And yeah, that's it's pretty interesting. So it seems like lactose tolerance is based on a mutation in the regulatory sequence of the gene. So the enzyme required to digest lactose, it's called lactase. It's right here.
Yeah.
And the regulatory DNA sequence is upstream of that gene. So this is an example of a mutation that does not cause a change in the amino acid sequence, but it causes a change in the regulation, aka the expression of a gene. Are you following?
Say that one more time?
Yeah, so we were talking about mutations that change the amino acid sequence of whichever protein that the gene codes for, right?
Yeah.
So that's one type of mutation. Now, this is another type of mutation that occurs in a regulatory sequence. So here we have the gene for an enzyme that breaks down lactose. And instead of the mutation being in that gene, which makes the amino acid sequence for the protein lactase, the mutation is located in a regulatory sequence. And if the mutation's in a regulatory sequence, it doesn't change the amino acid structure of whatever that gene is coding for, but it changes whether or not it's expressed or the level at which it's expressed.
Oh, okay.
So, like in these regions, like where there's 100% lactose tolerance, which means that people here can digest milk. It's like this, this is like Sweden over here, like Norway, Sweden over here, and then I don't know what this country is in Africa. But in these two regions, right, we have the regular DNA sequence is GC, but there's a mutation in these regions which turns it to CT. And that means that it basically like turns on the amount of expression of the lactase gene. So they're making a lot of lactase so they can digest milk.
Got it.
Yeah. So they're saying some mutations don't lead to a change in the amino acid, but it changes the level of expression of a gene. So 10,000 years ago, there was a mutation that allowed lactase to be expressed in adults, not just in infants, infant to 3-year-old people. Because originally human beings we evolved to only be able to digest milk from like infant age to 3 years old, since that's when we are breastfed. But then we're weaned off. So as adults, we're not really supposed to be digesting milk. But 10,000 years ago, a mutation happened which allowed certain people to digest milk as adults.
I see, okay.
So individuals that can digest milk, the sugar, which is lactose, from domesticated cows may have an advantage during starvation. So that's like an evolutionary reason that this mutation persisted.
I see. Okay.
And so we're basically going through like these different types of mechanisms of mutation. So, we started with mutation within a gene. We just did mutation in regulatory DNA. And now we're going to talk about gene duplication and divergence. So you can see from this image, right? This gene over here gets duplicated. And mutations are in that duplicated version of the gene, which means that it's no longer a duplicate, but it diverges, it changes. Right?
Yeah.
Okay. Okay, now this can occur during the repair of double-strand DNA breaks, or meiosis. So in genetic recombination, aka crossing over, you have basically this. So on the left, it's like a schematic kind of drawing. So, within the nucleus, we have, a chromosome, and here there's a gene for eye color. Here there's a gene for an enzyme called phosphorylase, and here there's a gene for collagen. Now, during homologous recombination, when the homologous chromosomes line up during metaphase, and then genes can cross over, you have this thing happening. So, the red chromosome and the blue one, they're homologous chromosomes. They're lined up next to each other, and basically this, where they cross over, that's why it's called crossing over. Like here, and here, there's going to be essentially, if you think about it, for the DNA itself, the DNA is going to have its double strand broken and put into the other chromosome and vice versa. So here, that's why you have, one of the blue genes on this red one and the red gene on the blue one. Right?
Yeah.
Okay. Gene duplication / deletion by unequal crossing over during meiosis. So, here we have homologous chromosomes that are aligned, right? And, they call the blue ones, that they use capitals for the blue ones. So, capital A, capital B, capital C. And for the red ones, they just use lowercase. Lowercase a, lowercase b, lowercase c.
Mm-hmm.
And then, so this is when they're aligned, in metaphase. And then there's crossing over, so, they, literally overlap each other. And then they, trade, the genes at that location. So that's why you have this red portion, that capital C from the blue goes to the red chromosome, and the lowercase c from the red goes to the blue chromosome. Do you see that?
Yes.
Okay. And so we call them now nonrecombinant chromosomes for this kind of unequal crossing over. So here's a close. So this is what's happening on the chromosomal level over here. Do you see this? So this is what's happening in the chromosomal level, and on the DNA level over here, you basically have this happening. So you have, gene on these two homologous chromosomes. And then the red parts are short repetitive DNA sequences. And you basically have this misalignment of the two. So they're aligned over here, but they're not aligned here. See how they're misaligned?
Mm-hmm.
And that means that the crossing over is going to be unequal. Because in this diagram of the chromosomes, like, they're completely aligned. So it's equal crossing over. But here they're not aligned. So you have unequal crossing over, which produces essentially a long chromosome with a gene duplication. So see how it's essentially like the size of the two chromosomes put together?
Excuse me.
Excuse me 1 second.
Yeah.
I need you to print something out because I have to submit it. I sent it to your email.
You turn on the printer.
Yes. The next time.
Is that your mom? You can tell her I said hi.
Mm.
Hi.
Hello.
What you doing?
I'm good. How are you?
Okay. Glad to hear you.
Yeah, glad to hear you too.
You see it? I have your manager degree.
Yeah, that's good.
Yeah, I'm director of operations.
Oh. Okay. Sorry about that real quick.
Oh, yeah, no problem.
Ready?
Yeah.
So wait, I'm missing where they're misaligned. Is it because they're only matching on like one side?
Yeah. So like
And not matching
here,
on both?
here they're aligned, right? So you have the short.
Yeah, okay, because they're matching on both sides. I see it.
Mm-hmm.
Okay. I got it.
Yeah.
Then the unequal crossing over
Mhm.
will cause a shorter chromosome to be produced
And a longer one.
and a longer one to be produced.
Yeah. So you see how
And
this is the
that can
long one.
form a
What's that?
genetic mutation that can be harmful.
Sorry. Wait. What?
Can that form a genetic mutation that's harmful?
Yeah. It can form a mutations are like kind of neutral. They can be good. They can be bad. Like the
Mhm.
lactase one is good for people. Because if they're starving and stuff like that, they can drink milk. So they can be good or bad. This one, most chromosomal kind of mutations are on a macro scale because they affect a lot more genes and DNA, and especially if it's making like a really long one and a short one that are together. So it's going to affect a lot of things.
Okay.
Yeah. So, an example of this can be the evolution of globin genes. So, essentially we have... so here's an example of a good, this unequal crossing over causing a good, causing a mutation that is a good thing. So we have over here a single chain globin which binds to one oxygen molecule, and and then it evolves to have a second globin chain by gene duplication. Because the thing is, the unequal crossing over, that may be the case, but it makes the gene duplicated. It duplicates the gene. So on this one chromosome, you have 2 copies of that gene. Right?
Mm-hmm.
So like, that's how. So like, for instance, like globin is just one, gene that would code for that.
Yeah.
And then through duplication, it will make another one. So hemoglobin for us has 2 alpha subunits, 2 beta subunits. So that means that the original one got duplicated to make another subunit, and then it got duplicated yet again, and that second duplicate got mutated into a different version. So that's how it turned into alpha and beta. So that's how we got from this to this. So a hemoglobin, essentially, that we have in our blood can bind to 4 oxygen molecules. And then here's an example of this like occurring through evolution. I think you froze. Okay, you're back. Okay, so do you see this over here?
No, not frozen.
Hmm?
Yeah.
Okay, so starting from down here, because this is millions of years ago, so like 700 million years ago, the gene was just for a single chain globin. So for this blue guy over here, right? And then 500 million years ago, it basically copied, or a mutation occurred that turned it into alpha globin. Okay?
Mm-hmm.
So it kind of changed, the structure a bit. And the version of it that did not mutate to form alpha globin, around 200 million years ago, it formed beta globin, the beta subunit. And there's a fetal version of it, which, the fetal hemoglobin, it has like a stronger affinity for oxygen, and an adult hemoglobin. And all of these are found on chromosome 11. So you essentially have, like, from just this single gene, you have a mutation that causes a duplication here, and then a mutation that causes a duplication here. Do you see that?
Yeah.
Okay. Chromosomal localization of a duplicated gene. So basically have them here and here. Alright, now this is the 4th way that genes and genomes can be altered. And that's through exon shuffling. So essentially, we have gene A here, and the green ones are introns, and gene B here, and the red ones are exons. And the exons can move around. So the exon, like one of the red ones, goes up here next to gene A, and Yeah. So, homologous recombination between exons. So here's the original genome. You have gene A here, gene B here. And you have introns here, exons here. And they get shuffled, so one of the exons that was originally near gene B goes to this point, near gene A. So the correspondence between exons and protein domains. So part of a protein sequence that can function, evolve, and exist independently of the rest of the protein chain. Usually 25 to 500 amino acids in length. Multi-domain proteins probably evolved by the fusion of genes that once encoded separate proteins. So here's an example, right? So in DNA over here, we have exon 1, which is like green, exon 2, which is like peach colored or whatever, and exon 3, which is like kind of orange. And they go on to produce a polypeptide chain with 3 different domains, one for each of those exons.
Okay.
Yep. Okay. Exon shuffling can generate proteins with new combinations of domains. It has been proposed that all of the proteins encoded in the human genome, there's around 21,000, arose from duplication or shuffling of a few thousand distinct domains of around 30 to 50 amino acids.
Okay.
So here we have EGF over here that has this green kind of like hexagon here. Chemotrypsin, which has this like brown kind of like oval thing here. And then urokinase, which has the green hexagon, the brown oval, and a new thing, blue square. And then factor 9 has, the 2 green hexagons, the brown oval, and then this yellow triangle. Plasminogen has the brown oval and 5 copies of the blue square. So essentially what you're seeing is essentially this guy gets duplicated a couple of times to make this or that. And then some of it gets shuffled to move around. So you basically see how all of these proteins are like just basically versions of these like simpler ones, right?
Yeah.
Okay. An exon can be duplicated by recombination. So we basically have 2 homologous chromosomes, exon A, dark blue, exon B, light blue. Here's a misalignment happening over here. And unequal crossing over. So, it makes a long one and a short one. And, now on that single chromosome, homologous chromosome over here, the single long chromosome over here, you basically have a duplication of exon B, because you have 2 copies of exon B, right?
Yeah.
Okay. Let's see. Genes and genomes can be altered, but okay. So now we're going to talk about transposition. The evolution of genomes has been accelerated by the movement of mobile genetic elements, jumping genes. This is around 45% of the human genome, hundreds to thousands of base pairs long, copied and inserted into a new site in the genome by the process of transposition. Can be half to 1 million copies. Sequences appear to serve no other useful function. 99% can no longer move due to mutation. Can generate mutations. 1 in 500 human diseases. Wait, what? 1 in 500 human diseases causing mutations. Okay. So here is an example for DNA only. So they're called transposons, okay? They're mobile parts of the DNA that can move around. And there's two types, cut and paste and replicative, which means like copy and paste, basically. So here's an example of cut and paste transposition. So the transposon is this thing in the red, and this is the target DNA, and it moves from this donor DNA to the target DNA. And then this is replicative, which is like copy and paste. So basically, this red transposon gets copied and pasted into the new DNA sequence. And it's copied, not cut, because it's in both the donor DNA and the target DNA.
Sorry, it's just moving really fast for me right now at the same time.
What's up?
Sorry if I'm taking a while. It's just I'm trying to copy this down as fast as I can. It's just a lot to take in right now. Kind of thing. All right, ready.
Okay. So, so transposons, there's a cut and paste one, and there's a copy and paste one, basically.
Okay.
All right, and then the enzyme that catalyzes the transposition is called transposase, and the most common genes in eukaryotes are actually transposases. The transposon DNA encodes the transposase. It's common in bacterial DNA, apparently. So, like, here's an example of transposon DNA, and transposase are these green, like, spheres over here, they bind to the transposon DNA, and they basically cause like a cleavage to occur, and then it can move to the target DNA. So it's target capture and integration, where it integrates the transposon DNA into the target DNA.
Okay.
Okay. Does this make sense? The transposon stuff?
Yeah, it seems to be pretty straightforward.
Okay.
Yeah, it's just cut and paste. Or
Copy and paste?
yeah. Yeah, the cut and paste where they just take one transposon and then throw it into another target DNA.
Mm-hmm.
Fine. And the replicative one is it's just gonna make a new one and there's two
Yeah.
instead of just one.
Yeah. So you can think of that as copy and paste.
Yeah.
Alright. And then transposases are the enzymes that like move that transposon DNA to the target DNA?
Yeah.
Okay. Okay, transposons can create new exon arrangements.
Wait, transposase moves the transposons, right?
Yeah. It's it's the enzyme that actually does that.
Okay, got it. Next.
Alright, transposons can create new exon arrangements. Exons move to another gene. So we have gene A that contains two similar transposable elements in introns. So, the exons here are the ones in blue. The gray are introns, and then the transposons here are the red ones that are inside of the gray. So they're inside of the introns. Okay, the transposase, that enzyme, it recognizes the ends of two separate mobile elements, aka two separate transposons. So it recognizes the ends of these. Like, the ends are like this and that. Like those, right?
Yeah.
Now, improperly excised, which means cut. The improperly cut transposon carries a fragment of gene A, which includes one exon.
Okay.
So it basically recognizes this end and this end, alright? And it like cuts these out and connects them.
Yeah, to make them into one exon.
Yeah, into one, like, essentially like transposon that is between, yeah. Because normally it would be taking out, like, it would recognize these two ends and take out this entire transposon, but it incorrectly recognizes the end of one transposon and, like, I guess, like, the beginning of another one and treats them as two ends of a transposon. Like, here, I'll draw this to make it make more sense. So, like, basically, that's like transposon 1, transposon 2, and, you know, basically so, like, you know, that's, you know, the start, I guess. That's the end. You can see what I'm drawing, right?
Yes.
So the transposase, it recognizes, you know, that, and then it, you know, it cuts it out. So, like, you know, let's say this is the new one, and then, you know, it puts it there, right?
Yeah.
Okay, now, what can happen, though, is it can recognize this start and this end and think that this is a transposon. Right?
Yeah.
And so, you know, let's say this is, you know, exon 1. It can make an incorrect transposon, which is basically, you know, that.
I see, okay.
So, you know, it's supposed to recognize, you know, the start. It recognizes the ends of a transposon, but, you know, here we have transposon 1, transposon 2 with an exon in between, and it can incorrectly take the end of transposon 1 and the start of transposon 2 and believe that to be the transposon to create this over here, where an exon moves as well.
Okay.
Yeah, so the improperly excised transposon carries a fragment of gene A, including one exon. So that's what we have here, because it just cuts this thing out.
Yeah.
Yeah. And now this is the transposon now, and this is the target gene, which is normal gene B. So, you know, it just has exon, the gray stuff is introns, right? And it inserts the new transposon into gene B. So it takes that and it just shoots it into this over here. So that's what we have there. So now the new gene B contains an exon from gene A.
Okay.
Yep. So that's just an example of transposons creating new exon arrangements. So we already know about like alternative splicing, but now we know that transposons can also essentially create something like that.
Got it.
Alright, now let's see. RNA-mediated retrotransposon pathway. This is unique to eukaryotes. So let's see. We have donor DNA with a retrotransposon. Okay. Transcription by RNA polymerase, which turns it into RNA. Reverse transcription to form single-stranded cDNA, which is this. Conversion back to the double-stranded DNA, which then is inserted into the recipient DNA. And then you have the recipient DNA with the retrotransposon.
Okay.
So this is just a way for you to take the end product of a transposon, like, you know, where you take, so a regular transposon starts with a donor DNA and then it moves that to the target DNA. This is a way for us to do it with the end result of the transposon process to put it back into a different one. So you basically take the donor DNA with a retrotransposon, you transcribe that into RNA, you reverse transcribe it into single-stranded cDNA, let that become double-stranded, you put it into the recipient DNA, and then you have, you know, both the donor DNA with the retrotransposon and the recipient DNA with the retrotransposon.
I see, okay. So essentially, it does the same thing except it throws it back to the donor DNA as well.
Oh, so this is like basically kind of like the copy and paste mechanism. Yeah.
But except it's coming back to the donor as well.
So it never leaves the donor. So this is like basically whoops, this over here. Because like in cut and paste, it leaves the donor. Here, it makes a copy of itself, and then so it never leaves the donor. It makes a copy of what the donor has, and then it takes a copy and it puts it into the target DNA. Okay. So yeah, and you make the copy by transcribing it into RNA, and then cDNA.
Oh, okay.
Yeah.
Got it. So, in order to get it to in eukaryotes, the way to get it there is to make it an RNA polymerase, I guess, so it's small enough, right? And then transcribes, reverse transcribes, and then they can convert fully into DNA, and then it throws it in.
Yeah. So this entire thing over here.
That's how they can get two.
Yeah, so this over here, so it doesn't take it out of the donor like you saw in the previous ones. It makes a copy of what's there. And then it takes a copy and it puts a copy in.
Yeah, I see. Right.
All right, and I guess these are just examples. 15%. Okay.
Yeah.
Oh, all right. So they talk about it. So line 1 elements, 15% of the genome, around 6,000 base pairs long. The mysterious jumping gene that appears 500,000 times in human DNA. Its segments make up 17% of our genome, but scientists are just starting to understand what it does. So line 1 RNA needed for mouse embryo development. The RNA acts as a molecular glue to assemble molecules needed to leave the 2-cell stage. Blocking the jumping ability did not affect embryonic development. Embryos may be able to make line 1 RNA, but prevent them from jumping in the genome. Okay? Alright, genes are... So I guess this is just emphasizing how these transposable elements are just a huge part of our genome. Like, this is like, whatever, 17%, 15%, 10%. And in comparison, genes, not transposons, like actual genes that code for a protein, they're sparsely populated in the human genome.
I see.
So, for yeast here, you see like a ton of genes, all this orange stuff.
Yeah.
In a fly, there's still going to be a lot, but you have introns and exons because flies are eukaryotes. But in humans, the genes there's just one here, 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 and here. Most of it is, not genes. So here, DNA segments of 50,000 nucleotide pairs contains. So for a yeast, there's 26 genes. For a fly, 11. For humans, only 4. So, and this is the last one for this PowerPoint. Most of the human genome is made of non-coding and repetitive sequences. So, you got lines, signs, retrotransposon, DNA only transposon. All of these are mobile genetic elements. We got repeated sequences, which includes all the mobile genetic elements, along with just simple repeats and segment duplications. And then let's see, unique sequences. So non-repetitive DNA that's in neither introns nor exons, and then a ton of introns, and then just this red part is the only stuff that are genes that code for actual proteins, and that's tiny. So the average gene is 27,000 nucleotides, but only around 1,300 nucleotides in exons. Only around 1.5% of DNA in protein coding regions, and then 3.5% are conserved with other mammals that do not encode for protein.
Got it. Okay.
Yeah, I'm gonna use the bathroom real quick.
Alright.
Okay.
Alright, so you want to go over the test?
Mm-hmm.
Alright. There's one with no answers, the first one.
Yeah, let's go over the one that says Chapter 9 genome questions first.
Alright. Yeah, there's only like 7 that they put here. Sadly. But that's probably the amount that's going to be on the test anyway.
Wait, what?
There's only like 7 different questions for it.
Yeah, I see 9, but...
I'm saying it's not like a long section. But that's how long it would probably be. Alright, so let's go over it.
Okay. Alright, the first one... a scientist uses a specific probe for a gene to label human metaphase chromosomes using fish. If they see 4 fluorescent spots, this likely means that this gene exists as how many copies in the haploid genome?
Would be 1.
So why is that?
Well, obviously it says that it's on there.
Yeah.
But, what do you call it? Isn't that supposed to be like where the genes are located on the chromosome?
Well, how do you get from... Well, yeah, like when you use fish, you're basically tagging some type of part of the gene that you want. But why
Well, diploid is 2, so haploid is 4.
Mm-hmm.
So what do you call it? It would obviously show the 4 fluorescent spots.
Wait, say that again?
I'm saying, isn't diploid 2 haploid 4?
Well, diploid is like 2. Haploid would be 1 though. Like in a sex cell, it's haploid.
Oh
you
wait,
have 23.
yes, sorry. Oops.
Mm-hmm.
Oh, sorry. Okay, but why would it be 1?
So we got okay, so 1 copy in haploid, 2 in diploid, but they're saying that these are human metaphase chromosomes. And in metaphase, you basically have, the homologous chromosomes, like, lining up, right?
Yeah.
So, that's why you're going to have 4 of these. Because you have... So these are... So that's, diploid. And that also is diploid. And diploid, so like, haploid is like 1N, N being the number of chromosomes. So for humans, N is like 23, right? Diploid is 2. And if you have 2 diploids, right, you would just get 2 times 2N, so 4. It's because you have homologous chromosomes.
Oh. Okay, now I get it.
Yeah. Yeah, so that's why you have 1 in the haploid genome.
Got it, because it's on 2 homologous chromosomes.
Yeah, and yeah. So and then
Well,
each
1 homologous like
Yeah.
chromosome.
Yeah, like
So
pair.
4 different locus.
Mm-hmm.
That's what it is. Okay, so 4 different locuses. Okay.
Oh yeah.
All right.
Exactly. Good.
That's what it's locating. Got it. Okay. That
Yeah.
makes sense.
So that's what they say here. Why are there 4 dots? Okay. Okay, single nucleotide polymorphisms.
Oh, okay. Well, it's either, and that one makes sense.
Well, let's see. Genes that are single copy in the genome due to gene deletion.
No, that's not it.
Why wouldn't it be that?
Well, aren't SMPs what do you call it? Associated with DNA, mostly? Like DNA in itself, not genes.
Okay, good. What if it said nucleotides that are a single copy in the genome due to nucleotide deletion?
Well, that would still be false because it's supposed to be like a mutation's a change. It's not saying like it's a deletion.
I mean, there's mutations that are deletions.
I mean, yeah, true. Hmm. Yeah, but if you delete it, then you have nothing. There's nothing single.
Well, I mean, okay, so what if so I wrote here a DNA sequence, and then let's say we get rid of this to get this. I mean, that could happen.
Yeah. But isn't that still just a change? That's not a deletion?
Oh, so I deleted the one that I circled, the T.
Oh, you did. Okay.
So, I mean, that could happen.
Yeah. Okay, then I don't know.
So the thing is that when you delete a nucleotide, it changes the reading frame. So when, before the deletion, like, this is a codon, right?
Oh, yeah, okay.
And this is a codon.
Mhm.
But it changes the reading frame, so now this becomes a codon, AGC. And then
Yeah.
the ATG is the next codon. And that changes the structure of everything that comes after it.
Yeah.
So in a single nucleotide polymorphism, all you have is a replacement.
Got it.
So like a substitution.
Yeah.
Okay. So let's see. B says the mutation that causes the single amino acid substitution in individuals with sickle cell anemia. So now we have, single amino acid substitution, which means that the codon, essentially like something changes. Because a single nucleotide is changed, it's substituted with another one, and now it's going to make a different amino acid.
Okay.
So that's an example of a single nucleotide polymorphism.
Okay.
All right, let's see. single base changes between one portion of the population and another. So that's also going to work because it's a single base change.
Yeah.
Good.
Exactly. So that makes sense. Okay.
What would this be though?
Which one? The driving force of transposon movement?
Mm-hmm.
Transposase?
Good. Okay. Alright, so both B and C are good. Alright, correct. Good.
What makes some people lactose intolerant as adults? Let's see. So some people don't produce as many enzymes, including ones that need to digest lactose. No.
What's wrong about that?
That doesn't make any sense because everyone produces enzymes.
Okay, good. The word any is what makes that wrong. Good.
Yeah.
What about B?
Some people don't make enough lactic acid to digest lactose. Lactic acid isn't involved in the digestion of lactose. It's supposed to, that's like part of lactic acid fermentation, so that has to do with the muscles.
Mm-hmm.
The active site of the lactase enzyme can vary from person to person. That wouldn't be true either because active sites would have to be the same.
Good.
And D, a mutation in the regulatory DNA of the lactase gene of affected people. That wouldn't work either because, let's see, mutation in the regulatory DNA of the lactase gene. Well, then if that was the case, then that means they wouldn't have been they would have been lactose intolerant totally, no?
It would be lactose
From the time that they were
tolerant,
young.
right?
Yeah, they would just be lactose intolerant from the time they were young.
Oh, wait, no, oh. Well, this would make them lactose tolerant, meaning that they could...
Oh, yeah. Oh, yes, that's right. Yeah.
Mhm.
Alright, never mind. Yeah.
Yep.
Okay.
Alright,
So, yeah, none of them are correct.
good. Alright. Gene duplication is thought to occur by... So, okay, what's a nonsense mutation?
It's basically a mutation that has nothing to, like, really do with the entire genome. It doesn't really affect anything.
No, it affects everything, which is why it's called nonsense,
Oh.
because it turns it into nonsense. But it basically is one that causes a premature stop codon.
Okay.
So, like, remember, a stop codon is, like, the codon that, during translation, it stops the building of that polypeptide chain.
Yeah. Oh, okay.
Now, if a stop codon is premature, like, some mutation happens, so it's, like, earlier than it should be, like, it'll stop the formation of that polypeptide, like, way before it should be stopped.
Yeah.
So that's what a nonsense mutation is.
Okay.
So, yeah, if you have, like, are you able to, like, write on this document?
No.
Alright. So, yeah, like, I would write down that somewhere. Or, you know, you can, I'm sure you can, like, just open it up in something that lets you write on, like, a PDF or something, right?
Yeah, I have nonsense mutation written down here.
Oh, okay. If you write it, like, next to these, then it could help you in, like, when you review for your test.
Yeah. Hold on.
Yeah. Because what we're basically, like, doing is just, like, we're going through each of the choices, and I'm going to ask you, like, other questions, like, based on the choices, so we can get a lot of bang for our buck. Like, instead of being like, okay, we know the answer is C because it says it's C, I can ask you, like, you know, what a nonsense mutation is, you know, if they change the wording.
So what's a nonsense mutation again?
So it's basically, it's a mutation that puts, a premature stop codon.
Yeah.
So it makes the during translation, it makes the polypeptide chain that the gene is supposed to make.
Wait, so during translation, it makes the polypeptide.
Yeah. So normally, here I'll write something down here. So normally, right, there's a start codon, and then there's like codons for the amino acids, and then a stop codon.
Yeah, and then this one like make it stop in the middle.
Yeah.
Let's say, or right after it just started.
Mm-hmm.
Okay.
Yeah.
So it makes the polypeptide chain. So during translation, it makes the polypeptide what?
Oh, it makes the polypeptide chain like essentially like it stops translating it before it's complete. So it basically just makes nonsense. It makes like some nonsense like, amino acid like chain that doesn't really do anything.
Nonsense. Makes the polypeptide chain make a nonsense chain that doesn't really code for anything?
Yeah.
Yeah. Now that makes sense.
Mm-hmm.
Okay. Got it. So B, unequal crossing over of chromosomes during mitosis, that would be wrong because crossing over would only happen in meiosis, no?
Correct.
Yeah, because this is during gene making, so that only has to do when basically fertilization and all that stuff's happening.
Mm-hmm.
Exon shuffling, that wouldn't be bringing up any sort of gene duplication, but it can bring up some sort of mutation.
Uh-huh.
And alternative splicing, that has to do with mRNA.
Yeah.
That has nothing to do
Where you
with
get rid of the introns? Yeah.
Yeah.
So it's the unequal crossing over of chromosomes during meiosis.
Yeah. That's why I skipped it over and I was like,
Yeah.
The blank needed to move a DNA-mediated transposon is encoded for by a transposon DNA. Well, that's transposase, that's already known.
Yep. What's a reverse transcriptase?
So that's going to be used during, what do you call it? RNA-mediated, what do you call it? Transposition?
Yeah, so like what does it do?
So it turns the DNA into an RNA polymerase and stuff, reverse transcribes it, and then turns it into DNA to put it into a new.
So think about it like this. So like
replication DNA.
So okay, so during transcription, well, what's a polymerase?
Huh?
What's a polymerase?
Isn't it an enzyme that codes for a polypeptide chain?
No. So it's going to be something that turns like a monomer into a polymer. So it's just gonna.
Yes. Okay. Yes, monomers and polymers. That's right.
So it can be like an RNA polymerase will add RNA nucleotides to a growing RNA strand. A DNA polymerase will do the same thing. It'll add DNA nucleotides to a growing DNA, strand. So that's like the general definition of a polymerase. And
yeah, we'll just grow it.
Yeah. So transcription is turning what into what?
Well, what do you call it? RNA into cDNA?
So just regular transcription?
Well, hold on. I have reverse transcription to form single-stranded cDNA.
Yeah. What about just regular transcription?
Oh, well, that just, what do you call it? It turns into RNA polymerase, no?
Well, okay, so when you get from DNA to RNA, right?
Uh-huh.
This process is called transcription, right?
Yeah.
But it's done through, just like a transposase is the thing that takes a transposable element and, causes it to move. Transcription is done through the enzyme, polymerase. In particular, RNA polymerase. Because it's the enzyme that basically adds RNA nucleotides.
Yeah.
So, like, if you had just in replication, what does DNA turn into?
DNA?
Mm-hmm. So, what's the enzyme going to be for that?
DNA polymerase.
Exactly. So the polymerase is the thing that causes. It's the thing that makes that mechanism happen. It does the work essentially.
Got it.
Okay, so if transcription is turning DNA into RNA, reverse transcription would be turning what into what?
RNA into DNA.
Yep. So that would be reverse transcription.
That's what I was saying before.
You were saying just the regular transcription.
Well, what I was saying was that it turns DNA into RNA polymerase and then recycles it back into DNA to be thrown back into the other.
Yeah, so it doesn't. So DNA to RNA is the transcription process. It doesn't turn it into RNA polymerase. RNA polymerase is what moves like the things along. Like it carries like the It basically carries the nucleotides together. Like here.
Yeah.
This is like an example of this red thing is DNA and green is RNA. So, DNA to RNA is transcription, but the thing that does it, that actually does the work, is this blue thing, which is the polymerase. That's the thing that adds RNA nucleotides onto that growing RNA chain.
Alright.
So think about like the overall process for like replication, transcription, reverse transcription, and then the enzyme isn't like what it creates, but the enzyme is the thing that carries that along. So like this over here, it would be, reverse transcription, and the enzyme there is reverse transcriptase.
Got it.
And I know that you're thinking of that specific example, but reverse transcriptase is in HIV.
Oh.
That's why HIV, you can't make a cure for it or a vaccine for it, because essentially it can change it. It can change itself like infinitely.
Yeah, I know when.
Yeah.
It's unpredictable. Okay.
Yeah. And then in the example for this though, that you're thinking of, it's also used like to make like a cDNA library. But like the purpose of the cDNA is what, like, because, all right, I'll just tell you. So like DNA, right? Only like 1 or 2% of it is codes for stuff, right?
Yeah.
Like only 1 or 2% are actual genes. So if you're going to use like okay, so DNA, whatever, it'll turn into like RNA, right? And then you can so this is like through transcription and then reverse transcription to make cDNA. Why not just use the DNA? Why go through the step of transcription and then reverse transcription to make cDNA? Why not just use DNA?
Isn't it a more complex process to even do it within eukaryotes in general?
I mean, like, it seems simpler to just use the DNA, right? Instead of having to get it to be transcribed and then reverse transcribed. So like
makes it easier to code for.
So let me ask you this. So when you transcribe DNA into RNA, right?
Mm-hmm.
What do we do to make it the mRNA? Remember those 3 things?
Say that again.
So when we transcribe DNA into RNA, what do we do to make it into...
No, you gotta say that again, you broke off.
Oh, sorry. When we transcribe DNA into RNA, what 3 things do we do to turn the RNA into mRNA?
I forgot.
Okay, so let's say that this is the so this over here is the RNA over here.
Mm-hmm.
Maybe that'll give you like a bit of a clue.
Oh, removes the introns.
Yep. Because why? What is an intron? Like why?
Well, an intron is like it codes for something. So for mRNA,
you mean the exon?
Oh wait, the exon codes for something. Yeah, sorry. But the intron is basically like just there.
It's just there, right? So
yeah.
if the coding part of DNA is just 1 to 2% of it, when we turn it into RNA and then mRNA through alternative splicing, remember?
Yeah, alternative splicing.
And then we turn that back into cDNA, then we're only taking the 1 or 2% of DNA that actually codes. Yeah, exactly.
Yeah.
So that's
it makes it simpler.
Yeah. So that's why
it harder.
Yeah. So that's why when they talk about cDNA, it's like they would talk about it in a cDNA library. So if you had to like do work on research or whatever on some type of gene, right? You can download DNA, which is going to be like a huge file, or cDNA, which is going to be 1 to 2% of the size of the DNA, and it's just the stuff that matter.
Yeah.
All right. Do you remember the other 2 things that happened after alternative splicing? And remember, it's because RNA itself is very unstable. So you want to protect it.
Yeah,
You want to protect the 5' and the 3'.
yeah, it's escaped my mind.
Yeah. So it's just a 5 cap, and on the 3' it's a poly A tail.
Poly A tails, that's right. Okay.
All right. So yeah, those are the 3 things that separate just the RNA transcript from the mRNA.
Yeah.
Okay. So yeah, so now you know what reverse transcriptase does and what a polymerase is. What about an acetylase?
Seems like it'll probably be an enzyme that catalyzes the production of acetylcholine or something like that.
So, basically it's just something that adds...
Acetylation?
Yeah. It adds an acetyl group.
Yeah.
Now, do you remember where we talked about that? We talked about it a little bit when we were talking about, like, histones and stuff.
Yeah, no, I remember that.
Yeah.
Yeah.
So basically, when you acetylate something, you spread apart the histones more.
Yes, that's exactly what I was thinking. Okay, yeah. Yes, I was remembering the picture of it,
Yep.
like, getting bigger, and then I was like, was it adding more, or was it just separating? So then, yeah, okay.
Yep. And what's the significance of spreading out the histones?
Doesn't it make it, like, easier to, like, what do we call it? Histone, antihistone, let's say. Sorry, just getting the context back in order. So if you're spreading them out, spread the histone out, it makes it easier to get in the they're surrounded by these like little ball looking things, right?
Yep. The ball
so it
things
makes it
are
easier
the...
to get into them.
Yeah. So...
Yeah, spreading it out makes it easier to get into there.
Makes it easier for what to get into there?
Binding proteins?
Yep. So, like, transcription factor.
Yeah.
Which is essentially just, a polymerase, basically, for it to transcribe it into RNA and cause it to be the gene to be expressed.
Yeah.
Okay, good. A flipase, that's like an enzyme that just like takes like a gene or some DNA sequence and flips it around. I didn't see that in any of the slides, so I don't know the importance of that.
No, I think that it's there to just see what the student knows or not.
Yeah. And then, transposase, good. So, if it ends in an ASE, it's an enzyme.
Yeah.
And enzymes are just the thing that does the work for the overall process.
Yeah.
Alright, let's see. Which of the following is true about the human genome?
Let's see.
Okay. So this over here, half of the genome consists of genes encoding for proteins. What's that number really like?
It's like 1 to 2%.
Exactly. Half consists of introns. I mean, it's probably more than half.
Yeah, more than half would consist of it. And exons only make up about 1.3 nucleotides per. 1.3 thousand nucleotides per.
Oh, yeah.
So, it's not going to be that much.
Yeah. And then, let's see. Half the genome coding for ribosomal RNA. That's still that's way too many. And then mRNA is the same thing. This and this are the same, right?
Well, C and E.
Wait, what?
Right?
Well, this is for ribosomal RNA.
No, I'm saying C and E.
Yeah, what about them?
You're the one. You said that they're the same thing.
Oh yeah, no. Because mRNA, right, is just the stuff that.
Introns. Picking them out.
Well, yeah. So then these would be different, right? But A and E are saying the same thing. Because the genes that code for the proteins are the ones that we keep in the mRNA.
Are the ones that we what?
That we keep in the mRNA?
Oh, yeah, okay.
Yeah. So
And
like,
then how?
so basically a gene, you know, equals all the exons, equals, you know, turns into proteins.
Yeah.
Yeah. So A and E are saying essentially the same thing.
Yeah.
Alright.
So which of the following is true? So about half of the genome consists of sequences derived from the mobile genetic elements, transposons. Yes.
Yeah. So I saw them say something. I don't remember them saying the exact that it was half, but oh wait, no, here. Okay, yeah.
Yeah, there you go. 45% of the human genome is accelerated by mobile. It's literally the title of one of the slides.
Yeah, yeah. Okay.
Okay. The DNA sequences in the genome of any two humans differ by how many single nucleotide polymorphisms?
This is like, you just have to know it, I guess.
Yeah, honestly. So
there's no other way to.
1 SNP and 1,000 NTs. Yeah, only 1 out of every 1,000 NTs are different.
Oh, you know what can help you know that? The fact that
What?
humans are 99.9% similar.
Similar, so only 1 out of 1,000.
So 0.1 is like 1. Wait, no,
Human.
that's 1/100th. Well, all right, I don't know. Just I guess know the 1,000th part. Wait, am I wrong here? No. Oh wait, no. 99.9% is 0.999, and so 0.1001. All right, whatever. Okay, 1 in 1,000. Okay.
Yeah. Alright.
Oh wait, no, that is true. My bad. 99.9%, 0.1% is 1,000.
Yeah.
Okay. So that can help you if you don't remember this exactly.
Okay.
Alright, what does the term genome refer to?
Let's see. So all the proteins expressed in an organism. No. Set of genes turned on in an organism. That doesn't even make sense. Turned on. No. The set of information in an organism's DNA not found in transposons. No. No. Not at all. Both A and B are correct answers. They're totally not correct. Complete set of information on an organism's DNA. That's straightforward. Yeah.
Yeah, it's basically the entire thing.
Yeah. Isn't it like 40 gigabytes?
Yeah, I was looking for the file right here.
One. Yeah.
Like right
You
here.
download one entire human genome.
Yeah, I remember when I was working with this. This was it. HG19 contains all these chromosomes. Whoops. All right, whatever. But yeah, it's a huge file. Okay. But yeah, it's basically everything.
Yeah.
What would this be?
Which one? All the proteins expressed in an organism?
Yeah. It's gonna have the same OME ending.
Oh, chromosome.
Oh no. So it's gonna be
Right?
it's gonna be this.
Oh, protosome?
Oh, sorry, proteome.
Proteome? Oh, okay. Okay.
And then
Wait,
there's also
wait,
yeah.
hold on. Okay.
Yeah, and then if it was like all of the metabolic processes, what do you think that would be called?
What?
It'll just if it said if you just replace this protein word with like metabolic processes, that would be the metabolome.
Oh, we have never talked about that.
Mm-hmm.
Okay.
So you see how they just end in OME?
Yeah.
Okay.
Okay. Set of genes turn on in an organism, tissue, or cell is basically express genes.
Yes. Good. Because not all the genes could be turned on, right?
Yeah.
Good.
Okay. Well, next one then.
Study of the human... Yeah.
Huh?
No, nothing. I was just reading at the same time as you.
Okay. It's revealed that approximately half of the human genome is composed of, well, it's basically the same question that's up there, so transposons or transposon-like elements.
Yep. Good. And another name for this would be like a regulatory region or a regulatory sequence.
Okay. Protein coding genes, that would be what we talked about earlier.
Yeah, like 1% or whatever, 2%.
Yeah.
And this is like
ribosomal
RNA.
Yeah.
Alright.
Same thing. Promoter sequences are when DNA is being replicated.
Well, it could be. Well, it'll be for like when it's Yeah, like transcribed, like basically a regulatory sequence. So like something like here. Wait. Like yeah, here, like regulatory DNA over here.
Mm-hmm.
It's like upstream of the gene. So like
okay.
the promoter is like, where the polymerase will attach to.
Yeah.
It's where the polymerase will attach to and start, the transcription or process. Okay.
Okay.
Let's.
Well, I have the test that doesn't have the answer, so I just won't look at your screen. So I'll try my best to answer it, and then I'll look if I'm correct, or you just let me know.
Oh yeah, I mean, I'll have this one open.
Yeah.
Which of the following macromolecules would produce only one type of monomer after complete hydrolysis? So, hydrolysis is the opposite of dehydration synthesis.
Yeah, condensation.
Yeah.
Well, one type of monomer. It would be RNA, no?
Well, I mean, RNA and DNA are going to be pretty much like behaving the same way here. And I think by complete hydrolysis, I think they mean like not just breaking it down into like individual nucleotides, but I think breaking the nucleotide apart. So
my only other guess would be glycogen.
yeah, I think it'll be. Well, actually, I think it'll be that. Oh damn. Okay. Oh, because it's one type. Okay. So this is why. So, So, DNA will break up into, nucleotide, which would break up into which, three things.
Yeah.
Yeah, like, what's the nucleotide consist of?
SNPs. Okay.
Well, that's a single nucleotide polymorphism. When a
Oh, sorry.
nucleotide,
DNTPs.
So yeah,
Right.
but if we break that apart, what are the three things that make that up?
I don't know.
So here's the drawing of a nucleotide.
Mm-hmm.
So like,
Well, the protein.
no protein.
Oh, the promoter.
So, here, check this out. So, I didn't want to draw out the entire thing, but that's what I meant by this P with a circle.
Oh. Phosphorylation.
Here.
Phosphate. Oh, okay.
So, phosphate, pentose sugar. What does pentose mean?
It's basically it has like 5.
And then the nitrogenous base, which is like adenine,
Yeah,
thymine.
guanine,
Mhm.
cytosine, or thymine.
Yep. So DNA would break up into these separate monomers. And RNA would too, right?
Yeah.
And triacylglycerol, what about that?
That's a fat,
Mm-hmm.
which makes it hydrophobic.
But what would that, break apart into?
Oh, 3. That's 3 glycerol groups.
Close.
Don't tell me.
Mm-hmm.
One phosphate, right? Well, 3 OH groups connected to a phosphate backbone.
So there aren't any phosphates here.
Huh? Okay. Connected to a glycerol?
Yep. So that's what that is.
Okay.
So 1 glycerol.
3 OH groups. Got it.
Well, they both have OH groups, but it's 3 of these. What's that group over there?
COOH. Carbonyl?
Well, The carbonyl is just
hydroxyl.
the hydroxyl is just that, and then them combined.
Carboxyl.
Carboxylic
Acid.
Mm-hmm. So these are the 3 fatty acids.
Oh, 3 fatty acids connected to a glycerol. That's right. Okay. I kept trying to remember to the first page written in my notebook. We had talked about that literally in our first tutoring session.
Yeah. So, yep. And then polypeptides, I thought for a sec, because one type of monomer is just going to be an amino acid, but it's going to be different types of amino acids. Right? But glycogen is just going to break apart into what?
Sorry?
Oh,
broke off.
Sorry, yeah. So glycogen will break apart into what?
Like glucose and oxygen?
Just glucose.
Or just glucose, okay.
Yep.
Glucose.
Yeah. So that's gonna be it, because it just
That's
all
the single
gonna be the
monomer.
same.
Got it.
Yeah.
Okay. Alright.
Good.
Alright, so next binds to the promoter of a DNA gene. Promoter DNA of a gene. DNA dependent RNA polymerase? I would not think so, no. General transcription factor that adds to the poly A tail? No. Title binding protein? Now that sounds correct. I remember that with the picture. The Tata box and everything.
Yeah.
Yellow.
Apparently more than one answer can be right for this question. And so, okay. So yeah, it'll be that and another one.
And then SNRP stand for single nucleotide. P stands for proteins.
Yeah. Or no, I think maybe, well, I mean, a DNTP would be a nucleotide pull. Wait.
Let me see.
Oh, yeah, no, you're right, Ah, got it, okay. Yeah, SNRNPs are small nuclear ribonuclear proteins.
That's... Damn it. Okay, yeah. That was my next thing.
Yeah, so like the significance of that is, so the SNRNPs form the spliceosome, which is like that kind of big like protein kind of complex that allows the splicing to happen, which is like the exons getting spliced together.
Yeah.
Yeah. So, but yeah, it's gonna be that. And that.
That'll be what?
Oh, the polymerase, DNA-dependent RNA polymerase.
That sounded correct too, but I felt like that wouldn't have been.
Yeah. So.
All right.
This.
Yeah, but now that makes sense. That's right. Okay, never mind. I got it. I got it. And the TATA binding protein, obviously. Okay.
Yeah. So. Like the Tata one, like I didn't like actually remember, so good on you. But the polymerase part is always going to be like true. Like for instance, if it was like the Tata one happens to be the transcription factor for some eukaryotes. Yeah. But like, yeah, this would be true for like everything. But yeah, it's gonna be both of these.
Okay.
Alright, what about purines and pyrimidines?
Purines are characterized by blank. Purines are characterized by blank. Well, your purines are adenine and guanine.
Yep.
Your pyrimidines are cytosine and thymine.
Yeah, and one more.
And uracil.
Yeah. I usually remember it by pyramid. I think of something that cuts because of its shape, so I remember CUT. So, yeah.
I don't know. B?
Yep. Yeah.
It's B?
Mm-hmm.
You sure?
Yeah.
Okay.
Oh, wait, no. Damn it. Alright, no. It's A. Sorry. Purines have 2 rings. Pyrimidines have 1 ring.
Okay.
Yeah, here we go.
Oh, okay, and the biochemistry of it. Got it. Okay. That's straightforward. The phosphate group of a nucleotide is usually attached to the... It'll be the 5 end, right?
Wait, I'm just trying to think of like a way to remember the purine pyrimidine thing. I don't know. What would be your way?
Well, pyrimidines, you basically think pyramid, and then you can basically fit like maybe 1 circle in there. I don't know. And then oh well, no, never mind. It's alright. That'll be fine to remember.
Okay.
Now number 4, I definitely think it's 5 carbon. Just from all that we've talked about.
The phosphate is usually attached. Yeah. So what would be what would be at the 3 end?
Poly A tail.
Well, that's poly A tail is just part of like the thing that's okay. It does add to the 3 end, but on the actual like molecule itself, what would be on it? Because it comes from this, right?
Yeah.
And the numbers refer to the pentose sugar.
Yeah. Oh, okay. Well, then the 3 end would hold maybe the phosphate.
Oh well, the 5 end would have the
Oh, okay. No, never mind. Sorry. The nitrogenous base.
that'll be on the 1 end.
Okay. So then the 3 end.
Oh wait, I messed this up. Hold, 1 sec.
I don't know.
Sorry, I forgot that there was another carbon there. So this is 1, 2, 3, 4, 5.
So the 3 end OH group. I don't know.
Yep.
Oh, okay.
Yeah.
That was my first guess, and I went away from it.
Yeah. So that's basically the most important like part, because that's the one that nucleophilically attacks another one. So like basically, if I had another phosphate or basically another nucleotide, this is gonna like in polymerization, right? This OH group attacks and connects to that phosphate over there.
Got it.
So that's how basically that's how it makes the that growing chain.
I see.
Yep.
Okay. By this is easy, so it's just gonna be. That's general bio 1 first thing you learn.
What? Oh, 5?
Yeah.
So what's your answer?
E. That both
Yeah.
A and B are correct.
Uh-huh.
What gives the DNA molecule its negative charge?
Yeah, that's a good question.
Um,
Oh man, I just remembered this.
no.
This is from like an Anki deck from like 2014. A and G are the big dogs. Purines, that is, and they're not afraid to throw their weight around. T and C are little bitches. They roll together for protection because they're smaller. Single ring. So I guess like if you think of A and G, all right, man. I don't know.
That'll be fine. I'll be able to remember it.
Okay. Yeah. So this is a good one. What gives DNA its negative charge?
It's probably gonna be that phosphate group.
Yep. Good. And histones are positively charged, so that's how DNA can bind with the histones.
Yeah. Their interactions are responsible for holding the nitrogenous base together. Stacked one on top of the other in the center of a DNA molecule and contribute to the stability of the whole molecule. Okay. Oh, wait. Okay, if we're talking about OH bonds, if we're talking about OH groups all over the place, then H bonds has to be a part of it, right? Or would it be van der Waals?
Well, let's see. So, if H bonds are true, that's a type of hydrophilic interaction, and I don't see any choice that says B and D.
Oh, okay, so it's gonna be van der Waals and then A.
Yeah.
Okay, got it.
Hydrophobic and van der Waals.
Yeah,
This is like the pi stacking thing. Did we go over pi stacking, or was that with someone else or something?
I think that was someone else.
Oh, yeah. Okay.
Yeah, they say it's both A and C over here.
Yeah.
That would make sense. I was just thinking H bonds because they would be like they're the strongest, but then I'm realizing a lot of stuff happens in DNA, so you wouldn't want to have a strong bond to that.
Mhm.
You would want a van der Waals reaction, which is just like regular.
Yeah. I mean, like, H bonds make sense because the bases, when they base pair,
Yeah.
it's through hydrogen bonding. And this is saying when they're stacked together, which is this thing called like pi stacking. So okay, so see this? So okay, you know, bases, whatever, base here, right? And then in the middle, right, between them, these are hydrogen bonds. But the stacking, right, because they're asking about holding the nitrogenous bases together stacked on top of one another. That's what is happening here. And that is this stuff over here. This, like, vertical kind of thing. Yeah.
Got it. Okay. The information encoded in DNA resides in. Well, isn't the phosphate backbone the DNA base sequence?
Well, the base sequence is the nitrogenous base, though, right?
Yeah.
So remember, nucleotide is composed of a phosphate, pentose sugar, and the base, like nitrogenous base, which does the base pair thing. Like the nitrogenous base is like what's going to be like ATGC and stuff.
Yeah. Alright, so then it's just going to be C.
Yes.
There won't be anything else.
Yeah.
Okay, let's see. Blank recognizes the stop codons UAG, UAA, and UDA. Release factors?
Yep.
Got it.
Because that releases that, you know, the tRNAs that make that, like, growing polypeptide chain. It releases it, and then the ribosome disassembles or whatever.
Yeah. What is chromatin that remains compacted during the interphase called? It is compacted densely stained chromatin is found at the nuclear periphery. Euchromatin?
Close?
Hetero, right?
Yeah.
Okay.
Euchromatin is the loose one.
Oh, okay. And then the compacted is hetero. Okay.
Yeah. And then the rest of them aren't anything.
Yeah. Of the following steps associated with translation, what does not involve hydrolysis of GTP? So.
Oh, okay. So this was like something that we kind of skipped when we came across it in the slides.
Yeah.
So I'll tell you what happens here.
Well, let me take a guess at least then.
Okay.
Translocation of the ribosome? or
Okay,
A?
it's apparently more than one answer.
Oh, maybe it's saying that it would accept either. Okay. Well, then I'm saying either A or D.
Let's see. One sec. Let me get one of my notebooks where I talk about this. GTP. GTP. Okay. In my notes, thankfully, I did each GTP thing. I circled it in red. Okay, so GTP is used in the first step where you basically where you make the initiation complex. Essentially, like when you put the two parts of the ribosome together to make like when you put the large subunit and the small unit and you put that together, that requires GTP. And then when you activate the tRNA, which is the amino acylation of the tRNA, that's also when you use GTP. And then when you translocate the like the tRNA from one site in the ribosome to the next site, that also requires GTP. Oh wait, one sec. Amino acylation. tRNA loading. Oh, got it. So that doesn't require tRNA loading driven by breaking high energy amino acyl AMP bond. Okay.
Alright, well,
Yep.
from the answers here, I got it right with A, but wrong with D. It's supposed to be C.
Yeah. So these other three things are the ones that require GTP. So I would like put that in your notes somewhere, and I can actually I can draw it for you as well, but yeah, this one, I don't know, highlight or whatever, just to know that, because yeah, that'll be important to know.
Okay.
Because yeah, when we first went over it, it was kind of detail that, that at the time for understanding the overall translation process wasn't big, but I can kind of include it.
Got it.
Okay.
An unusual structural stretch of heterochromatic sequences at the tips of the DNA molecule forming a cap at each end of the chromosome is called a carriomere?
I don't even know what that is. I don't think it's anything, to be honest.
What do they say here? Oh, they say it's a telomere.
Yeah.
Rip.
So what should help you is it forms a cap at each end of the chromosome.
Cap. Yeah. All right.
Mm-hmm.
I looked over that. I answered it too fast.
Yeah, and then heterochromatic here isn't what they mean in terms of heterochromatin over here. Or actually, no, that is what they mean. But it also comes from like how it's stained in terms of color. But yeah, what do you think it means, like that it's heterochromatic?
It's compacted.
Which means it doesn't get expressed, right?
Yeah.
All right, yeah.
Then 13 is obviously centromere. That's known.
Site of indentation on the surface that the
Mitotic spindle attaches.
Okay, yeah. What's the centriole?
Centriole is in that's a microorganism. That's a microorgan in itself.
An organelle?
Organelle, yeah. Sorry.
Yeah. It's like the thing on the ends.
Yeah.
Yeah. And the centrosome.
Same thing. It's an organelle in itself.
It's like the thing that. So like, for whatever. This guy, like, the centromere is here. And then I think the centrosome is like this, kind of the protein thing over here with the kinetochore and stuff.
Yeah.
That, it attaches to.
Oh, okay. Got it.
Which two monosaccharides combined form lactose?
Monosaccharides. So we're gonna do, I think, glucose and fructose.
All right, so that one is actually sucrose, which is like table sugar.
Oh, wait. Okay, no. Well, I'm close. If it's sucrose, that's sugar. So then it should be, and lactose is milk sugar. So galactose.
So it'll be glucose and galactose.
Oh, damn it. Okay.
Mm-hmm.
Okay, got it.
Yep.
rRNAs. So that's ribosomal RNAs.
Mhm.
Function, the informational storage capacity, are adapted to recognize and bind to other molecules, provide structural support, catalyze the chemical reaction equivalent. Just B, C, and D are correct. Let's see. Adapted to bind to other molecules, provide structural support, and catalyze. rRNAs can't catalyze anything because they're not a enzyme.
Well, remember RNAs can behave as enzymes?
That's true. Okay, so well, they are. B is true, right?
Yep.
So then it's gonna be E. B, C, and D are true.
Yeah. So basically, polymer from which ribosomes are constructed acts like an enzyme when assembled as a ribosome, polymerizes proteins. So basically, provides structural support, catalyze the chemical reaction that covalently links amino acids, and are adapted to recognize and bind other molecules.
Yeah.
Doesn't do anything for information storage.
Yeah. 16 transcription factors help RNA polymerase recognize promoter.
Yep. And you know the one that you mentioned was the Tata box.
Yeah.
Good. What does a protease do? Protease.
Enzyme that binds to proteins.
But what would it do?
They catalyze the production of a protein. Or the action of one.
Is it catalyzes the breakdown of one?
Oh, the breakdown of proteins. Okay.
What about a nuclease?
Characterizes the breakdown of nucleotides.
Catalyzes, yeah. Yep. So like that's why you need to put on the 5 cap and poly A tail.
Yeah.
To prevent it from getting broken down by nucleases.
Yeah, broken down more.
What's the difference between an endonuclease and an exonuclease?
So the endos are located within, the exos are located outside.
Yep. So the 5 cap and the poly A tail will prevent it from being attacked by exonucleases, but not endonucleases. Right?
17. Those portions of DNA succeeding following the initiation site towards the 5 end of the template are said to be blank that site. Downstream.
Yep.
Let's go.
Yeah. And yeah, upstream, that's where like the regulatory sequences and stuff would be. Like ahead of it. Good.
All 3 major types of RNA, are processed before they are mature. They are derived from precursor RNA molecules that are considerably longer than the final RNA product. These precursor RNAs are called I think just pre-RNA.
So, by the way, a nucleoside is the same thing as a nucleotide, except it's without the phosphate group.
Oh, so it's a nucleoside?
Oh, no, you're right. It's pre-RNA.
Oh, okay.
But yeah,
and not transcription factors.
What's up?
No, because they're saying there's an answer that has both A and B is correct.
Oh.
But
yeah. Transcription factor. Yeah.
That comes later when they're matured.
Yeah, that's like a straight up like even beyond that when it turns into like, you know, a protein and stuff, right?
Yeah. So ribosomal DNA clusters.
Do you know what prions are?
What?
Prion?
Prions? No.
So they're pretty interesting. They're basically like, we only discovered them recently. Like, you remember mad cow disease?
Yeah.
So that's done from this. So it's not a virus. It's not like a bacteria. It's a misfolded protein that can cause some type of like, that's a pathogen basically.
Oh.
Yeah. It's really interesting stuff because you see that in certain like, so like mad cow disease was when we were feeding cows. So
yeah.
And then also like in certain like tribes that are like cannibals, they eat human brains, and you can get a disease called kuru, which is prions basically. Yeah, they're super interesting to think about, because like they're really hard to kill because it's a protein.
RDNA clusters are gathered together to form the, I want to say nucleus. My gut is telling me heterochromatic body.
Oh, that's so this is just saying you know, well, in the nucleus you have DNA, but what about the rDNA? That actually forms another thing within the nucleus.
Yeah, I meant nucleolus.
Yeah, that's it.
When I was saying that modifications to the nucleotides of pre-RNAs are made pre-transcriptionally.
Well, the saying modifications, aka like alternative splicing, right? Adding the 5 cap.
Yeah, but to pre-RNA.
But yeah, so they're modifying the pre-RNA to turn it into. Oh wait. Yeah. Oh wait. Sorry. This is R ribosomal RNA. My bad. But yeah, if you modify any pre-anything, it'll turn it into like, you know, the mature version of it.
Oh, so post-transcriptionally then?
Yeah.
Okay. Ribosomes are made out of I think it's E.
Yep, proteins and ribosomal RNA.
One enzyme transcribes 5S rRNA. RNAP2.
So that's the one that is the major, you know, the thing that makes that adds the RNA nucleotides to the growing RNA chain.
So then RNAP3.
Yeah.
Okay. RNAP2 promoters are located on the blank side of each transcription unit. Then we got like time for like 2 or 3 more questions.
Mm-hmm.
RNA polymerase 3 promoters, showing 2 promoters are located on the blank side of the transcription unit. Ooh, terminals. Shit. C terminal?
Sorry, I was looking for like the definition of the different polymerases. RNA polymerase 2 promoters are located on the blank side of each transcription unit. Oh, okay. So, what did you think?
I said C terminal.
So N and C terminal are only on like amino acids or like peptides.
Hmm.
And RNA polymerase 2 will be on the like the DNA thing to transcribe the RNA.
Oh, okay, so then 5?
Yeah.
Okay.
Cause yeah.
The RNA sequence blank is called the polyadenylation sequence because the poly A tail invariably begins about 20 bases downstream from it. D?
So this one, I'm trying to see like how to like how you would figure it out, because I remember them talking about it. So I'm trying to see like how that answer would make sense, because it's.
I looked it up, but it's A.
Yeah. I guess. Well. Okay.
Okay. Figure that out. We'll meet again on Tuesday.
Wait, you don't want to go through this?
Well, the rest of this? Not today. I have some stuff to do.
When was your test again? Sorry, I keep asking that.
Next Wednesday.
What's that?
Next Wednesday.
Okay, on the 13th?
Yeah.
Okay.
I go in person.
Yeah. So
I think Tuesday's a good day to meet.
yeah, we could do that. I just want to take a look at because the questions seem to get a little harder. Okay. All right. I'll see you on Tuesday then.
Yeah, Tuesday's cool.
All right. Take care, man.
All right, take care.
All right.
Bye.
All right. See ya.
See ya.